So my presentation today is on DNA replication, so DNA replication by Emily Nettles. And the main question that I wanted to discuss today was what is DNA replication and what type of processes does it undergo? And the unit objectives that I wanted to cover was Unit 7.3, and it's outline the key steps in bacterial chromosome replication, create diagrams, drawings that show what proteins and DNA features are associated with the processes of initiation, elongation, termination. So why replicate? DNA replication occurs in order to generate a new set of cells in bacteria. This is done by binary fission, and information is transferred from one DNA molecule to another. Overview. So DNA replication is semi-conservative. Each new double helix is composed of one parental strain and one newly synthesized strain. And in this, we're going to be talking about something called a replication bubble. That'll come up below. So there's these conservative, semi-conservative, and dispersive strains that are talked about. So in the first replication in your conservative, it's one full of this type of DNA, and then of a different type of DNA here. Fully conservative, and then your semi-conservative is one strand has that type of DNA and the other strand has the other. So, and then your dispersive is not very important. So proteins involved in DNA replication are your DNA A, which is your initiator protein, which binds to the origin, your DNA B, which is helicase, and that unwinds the helix to expose template strands, your DNA G, which is primase, and that synthesizes RNA primers, and your DNA polymerase 3, which is your major replication enzyme, and then your DNA polymerase 1, which replaces RNA primer with DNA, and then your DNA diarase, which relieves positive DNA supercoiling. And those we'll talk about more later. So what are the processes? You have initiation, elongation, termination. Initiation, so DNA A melts the DNA at origin C and assembles a replication machine. That replication machine is called a replisome. The DNA C loads the helicase, which is DNA B. Then DNA B recruits DNA G, which is your primase. And primase synthesizes a short RNA primer against each template strand, and the replisome is completed by the addition of DNA polymerase 3. And that's held in place by a sliding clamp protein. And that's shown in this photo. It's just, here's this single strand binding proteins. Okay, so here's another photo of kind of all this happening in just like real time, so kind of more zoomed in. So you have your helicase loader with your DNA C, which is the helicase is here, right? And then your DNA primase is going to go here onto this leading strand, and it's going to be a primer for that leading strand there. In elongation, DNA polymerase reads the nucleotide sequence of a DNA template and synthesizes a complementary DNA strand. This requires a free 3' OH group, so new DNA has to be synthesized in this 5' to 3' direction. You can see that here in this photo, it's synthesized in this 5' to 3' direction. You have your leading strand, which is continuous towards that replication fork, and then your lagging strand, which has those Ozaki fragments, which are discontinuously synthesized, short DNA fragments. So your RNA primers are removed by RNase H, or the exonucleocase activity, and your DNA polymerase 1 then synthesizes the DNA patch using the 3' OH ends, and DNA ligase repairs the phosphodiesterase. So in termination, you have two replication forks meeting each other at the termination sites, and there are termination sites on the E. coli chromosomes, and there's also terminus utilization substances that bind to these sequences and ensure the polymerase complexes do not escape and continue replicating. And there's a photo of that. And then I now have some review questions. So what is the role of DNA helicase during DNA replication? It's unwinding the DNA double helix. DNA helicase unwinds the double-stranded DNA by breaking hydrogen bonds between base pairs. Which enzyme is responsible for synthesizing the new DNA strand during replication? It's DNA polymerase. DNA polymerase is a primary enzyme that adds complementary nucleotides to the growing DNA strand. What is the purpose of RNA primers in DNA replication? It's to provide a starting point for DNA polymerase. So DNA polymerase can only add nucleotides to an existing strand of nucleic acid. RNA primers synthesized by this primase provide the initial nucleotide sequence. And then in what direction does DNA synthesis occur on the new strand? It's the 5' to 3' on that new strand as DNA polymerase adds nucleotides to the 3' hydroxyl group. And then what are Ozaki fragments and where are they found? And they are short DNA segments synthesized on the lagging strand. Thank you for watching.